All right, so let's jump right into this. Start recording. We're good. All right, so the last chapter, chapter 16, is our special senses chapter. Okay, so we're going to talk about each of the special senses. You know, vision, all right, smell and taste, and we'll talk about some touch. But in order to do that, we've got to basically talk and get into a little bit of detail about the sensory portion of it, okay? Because that's what these senses are. It's sensations that we can consciously perceive, all right? And then we process it. So we're going to, before we get into that, we want to first talk about some of the special senses, or sensory receptors that are out there, okay? So to break it down into the easiest terms, the most broad category for our sensory receptors are going to be the ones that are going to be monitoring information outside of our bodies and then monitoring information inside your bodies, all right? Both your external and internal environments, okay? All receptors, all right, are going, let me rephrase that, okay? You need a stimulus for a receptor to respond to something, okay? So your receptor is only going to respond as long as a stimulus is present, okay? So your auditory receptors, right, I can flash a flashlight in your ear all day long, okay? You're not going to be able to hear the lights, all right? That's not the right stimulus, okay? So if I were to snap my fingers, then your auditory receptors would pick that up, okay? So my point being is we need a stimulus, all right, that is going to activate the receptors so we can move into that reflex arc, whatever type of reflex arc that we're getting into, okay? So my next point, talking about me flashing that flashlight in your ear, all right, each type of receptor, all right, is best suited for a specific type of stimulus, right? Obviously, sound means nothing to my photoreceptors in my eye, okay? So certain receptors, all right, respond, all right, for certain or specific stimuli, okay? So now, when we have that stimuli and that receptor, all right, is stimulated by whatever that stimulus is, we need to convert that signal, all right, that sensory signal, all right, into an action potential. So we have this structure here, which we call transducers. So they're going to convert whatever that stimuli is, all right, into electrical energy, otherwise known as an action potential or an AP, okay? Some sort of nerve signal, okay? So these receptors, we learned this concept already. These receptors have a resting membrane potential. So like when nothing is going on, all right, a lot of these receptors are just minding their own business. And then the stimulus occurs, all right, and then we reach that threshold value, and then we're going to conduct a nerve signal, an action potential, all right? So this breaks down into this concept here, that modality-gated channels. All right, modality-gated channel is chemical-gated channel. That's what that modality means, all right? Or an electrical-gated channel, voltage-gated channel, all right, pressure-gated channel. Please stop. All right. I hate hearing myself, okay? So that's what we're going to talk about. We're going to stimulate, and we'll talk more when we get into the specific senses here. Probably not today, but we'll get into that, all right, which gated channels are going to be involved here, okay? But ideally, we're going to generate these action potentials that are then going to go to our central nervous system and go to that specific lobe for whatever that specific sensation is. And then we'll interpret it and we'll figure out what that is. What's that sound? Or do I recognize that person? All right, what's that taste, you know, or that smell, okay? So that's what we're going to talk about. All right. So when we talk about sensory receptors, all right, like touch, for example, all right, we have to talk about the concept of the receptive field here. Okay? So the receptive field, okay, is going to be the square footage or square inches, whatever you want to call it, all right, but the real estate in which these receptors, all right, are going to be able to monitor, okay? So if you look at the picture here, you see that this guy here is a small receptive field, and then this one here has a larger receptive field, all right? The concept that you need to understand for a receptive field is, like a dermatome, that has a large receptive field. Okay, remember that dermatome map with all those lines drawn on the body there? Okay, but specific nerves now, and I know we briefly only talked about a few, okay, like the, what's an easy one? Axillary nerve. All right, we briefly talked about that. That's a nerve that comes off of the brachial plexus. And the axillary nerve, its receptive field is this little patch of skin on your shoulder here, okay? But your shoulder, all right, when you're in the anatomical position, all right, your shoulder, that dermatome is going to be the C5, C4 dermatome, all right, but that's a large receptive field, all right, so there's a difference. So basically, the concept that I want you to understand for receptive field, the smaller the receptive field, all right, the more distinctive, all right, or definitive the receptive field is. The larger the receptive field, all right, the more general it is, okay? You won't have as fine a touch for a large receptive field compared to a small receptive field. Small receptive field, more precise. You can distinct it. There's a sensory test that you can do. You have the patient close their eyes and you take two, I'm not going to say two sharp objects, but kind of two pointy objects, and you put them close together, right? And you like these two pipettes. Okay, you have two pipettes. You put them together and you tap the patient on the skin. And you say, "What do you feel?" "I feel a point on my skin." I said, "All right." And then I move them away just a little bit from each other. And I push them down at the same time. "What do you feel?" "I feel 1 point." And then as you move them further away and you push down, "Oh, I feel 2 points." All right? That's how you're able to determine, all right, the receptive field. Certain areas of your skin are going to have larger receptive fields than others, and some are going to have smaller receptive fields. I wish I could remember the name of the test. I can't remember the name of the test off the top of my head. All right, huge concept. Every semester I ask this. What is a sensation? All right? A sensation is the stimulus that you are consciously aware of. Okay? So when you talk about a sensation, a cold sensation, then you are consciously aware that something is cold. It's a hot sensation, you are consciously aware that something's hot, sharp, dull, whatever, okay? But it is, you are consciously aware. Do not miss that on a test, all right, about what the definition of a sensation is, all right? You've heard me talk about this many times. For us to perceive something, all right, for it to come to our conscious awareness, okay, it has to be relayed through our relay center, the thalamus. Once it gets past the thalamus, then you have conscious awareness of it. What's past the thalamus? The cerebral cortex. Any of those different regions there, okay? So, only a small portion of all the stimuli make it, all right, to the cerebral cortex, right? Think about it, all right? You're sitting in this room. There's sounds going on in this room. There's sensations that are, I shouldn't say, there's stimuli going on in this room that you're not even aware of, all right? Every time you take a step, you have all these sensory neurons just blowing up on the bottom of your feet, sending information up to your brain, all right? But you don't perceive a lot of that unless you step on something sharp, all right? Or you step in water and you're like, "Oh man, my foot's wet," all right? But there's tons of stimuli that are constantly bombarding your central nervous system, but you're only aware of a small fraction. If you were consciously aware of all that sensory stimuli, you go insane. You would literally go out of your mind. And that's one of the things that they talk about with autism, all right? A lot of people with autism, they don't have that filtering. So they are very, very sensitive, all right, to noises, specifically loud noises, crowded rooms with lots of conversations going on, freaks people out. I went to a wedding one time, and so we were on the dance floor, and there was a young boy there that had autism, and he just did laps around the dance floor. That was the only way that he could cope with, all the people moving, but he was fine. As long as he was doing that. You stopped him, boy, he would not be happy. Not be happy. Okay. So keep in mind, all right, so a sensation, conscious, perception, all right? So going back to what I was saying, all right, when we're talking about all these receptors, they're going to provide us, all right, information on the type of stimulus, all right, where that stimulus occurred, if it's a mild, moderate, or severe type of stimulus, how long that stimulus goes on, all right, and then the type of it, the modality of that stimulus. Is it a smell stimulus? Is it a taste stimulus? Okay, is it a pain stimulus? All right, so we're going to talk about those different things here when we talk about modality. Okay, the type of stimulus here. All right, so this kind of draws in chapter 13 when we were talking about, you know, association areas and higher functioning regions of the brain and whatnot. All right, certain regions of the brain are going to be designated for certain higher order functions. All right, perfect example, the occipital lobe is designated for vision. Okay, so when the photoreceptors in the eye are stimulated, they're going to send information via the optic nerve to the occipital lobe. And when those action potentials arrive there at the occipital lobe, then you're able to see. Right? Same thing with sound. All right? When those sound receptors, all right, are stimulated, all right, they'll change that sound energy into nerve energy, nerve signal energy, and then it'll arrive, all right, at the temporal lobe. And when those nerve signals arrive at the temporal lobe, then you'll hear. Okay? So again, it has to do with that type of stimulus is going to activate specific receptors, and those specific receptors are going to send sensory input to specific areas of your brain. And when they arrive at those areas, then you will be able to see, hear, taste, touch. Okay? All right. Let me go back one. All right. So that's modality. Intensity, okay, that's an easy one. All right. The more intense a stimulus is, the more frequent the action potentials will be. Okay? So keep that in mind. The louder the sound is, let's say, for example, all right, the more action potentials and nerve signals that will be generated in your inner ear. Brighter the light, for whatever reason, all right, the more action potentials will be generated in your optic nerve. Okay? So the stronger the stimuli, the more frequently the neurons will fire. Okay? And they'll just bombard, all right, your occipital lobe, temporal lobe, whatever lobe, insula, whatever, with that information, all right, for whatever that stimulus is. All right. Location of that stimulus, again, this goes back to the receptive field. Okay? That's more so for touch. All right? So going in the regards to someone touches you on the shoulder. Okay? Now you might not see them, but you feel somebody touch you on the shoulder. All right? You're going to immediately stimulate, all right, those touch receptors there in the skin of your shoulder. And it goes immediately up to the parietal lobe, right, where that post-central gyrus is. Remember that motor homunculus, that goofy-looking thing, drawing of your body and how your brain, all right, perceives your body, all right? It is going to go to a specific area, all right, of that motor, or the sensory homunculus. When it goes to that specific area, then you know where that is on your body map, right? That the fact that it's on your shoulder, because it went to that specific area of your sensory homunculus there. This one, adaptation, is always an interesting one, all right? Because have you ever noticed when you buy a new pair of shoes, sometimes everyone's like, you got to break them in, you got to break them in, you got to, you know, or you feel comfortable, or you put a pair of pants on or whatever, or a shirt on that might be too tight. But like an hour later, you don't feel that it's tight on you anymore, all right? And the reason why is you've undergone adaptation, all right? Those sensory receptors there have decreased their sensitivity, all right, to that stimulus, okay? So there's two types of receptors. There's tonic receptors and there's phasic receptors. The tonic, all right, have a limited amount of adaptation, okay? So that means that they're constantly going to be able to respond to the stimulus, all right? which is good. For example, our in our head, when you're turning your head from time to time, right? You don't get dizzy because the receptors there are constantly, all right, responding, all right, to the changes there, okay? So they don't adapt, so they're not decreasing their sensitivity, all right, to that ongoing stimulus that occurs. You wouldn't want that to happen with pain, that's for sure, okay? You hold on to a hot pan, what do? You let go of it, all right? But if you had, all right, the ability, all right, to adapt to that pain, that's bad. You could hold on to that pain. You're like, oh, I don't feel this anymore. Just give me 3 more seconds. I won't feel anything at all. You know, meanwhile, you're burning your hand. You don't want that to happen. All right. So pain receptors are tonic receptors. All right. They're slow to adapt. They will not decrease their sensitivity to that stimulus, especially if it's a painful stimulus. Basic receptors, all right, they adapt rapidly, which is good. Okay. You put that tight pair of pants on or the socks, brand new socks, right? And they're kind of restrictive on your foot. But once you put your shoes on, you walk around a little bit, you don't feel it as much, all right? Those are pressure receptors, all right? They respond quickly, all right? So you won't notice it as much, all right? So for a stimulus to be consciously perceived, the sensory input must be sent to the cerebral cortex. Normally when I do this, it's on a slideshow, so the answer doesn't pop up there. But unfortunately, this is not the case. Okay, so let's classify some of that sensory information, okay? So there's a couple ways to classify sensory receptors. All right, first is where we find them, okay? The next is the type of stimulus. And then finally, all right, well, actually, type of stimulus is last. The second one is where the stimulus occurred, okay? So for receptor distribution, there's general sense receptors and special sense receptors. How I remember the difference is if you go down to special sense receptors, all right, and we talk about our 5 special senses, all right, all of these, all right, are going to be in the head, okay? So special sense receptors are always found in the head. In the head. General sense receptors are found everywhere else, all over the body. Head and neck and feet and hands and arms, all right? Abdomen, trunk, everywhere. It's throughout the body. And we've talked about these type of these 2 senses here, the somatic sensory receptors and visceral sensory receptors, all right? Somatic sense receptors are going to be pretty much touch, all right, throughout your skin, okay? We include the mucous membranes, and then also, remember these guys, proprioceptors, right? And those are located in, of course, the joints, all right? What else is near a joint? Muscles and tendons, okay? So skin and proprioceptors for the somatic sensory. Visceral, remember, viscera, organs, okay? So the visceral sensory receptors are going to be found in our internal organs, okay? And so what do? Well, we talked about a couple things. All right, the rectum, for example, the stretch receptors in the rectum, the stretch receptors in the stomach. All right, so they're going to monitor stretch inside some of these hollow organs, small intestines, large intestines. The chemical environment, that's going to be in your bloodstream. If your blood is too acidic or too basic, for example, all right, temperature and pain, okay? All right. The origin of our sensory stimuli. Is it inside the body or is it outside the body? Okay? That's what we need to know. Okay? If it's outside the body, then we classify the receptors that pick that information up as exterior receptors. Okay? Skin, mucous membranes, and then our special senses. Sight, sound, hearing, taste, touch. Okay? If it's inside the body, think of the internal organs, which I just talked to you about. Stretch receptors inside of our internal organs, all right, is an example of an interior receptor. And then always I feel like proprioceptors, you know, they have their own classification. They're always on their own. And by now I hope everybody knows what the proprioceptors do, okay? They are going to monitor limb movements, body movements, okay? So how your arms and legs, where they are in position, all right, in relation to your body, okay? And these are located, and we talked about two types of two types of proprioceptors in chapter 14. All right, remember the Golgi tendon organs? Those are the proprioceptors that monitor excessive muscle contraction. And then we have the stretch receptors. Remember the muscle spindles? All right, they monitor how much a muscle is getting stretched. Those are two types of proprioceptors, okay? And they're all located in muscles, tendons, and joints. All right, and then finally, the last part of our classification for the sensory receptors, okay, is modality, the type of stimulus, okay? And so we've got 5 types. And it's a good idea if you kind of, well, you should always pay attention to everything I say, but if you really pay close attention, all right, to the chemoreceptors, okay, because you're going to visit those again next semester, okay? Chemoreceptors, chemical, all right, are going to monitor, all right, the chemicals in a dissolved fluid. Well, that is a lot of different things, okay, but most prevalently is going to be the blood, okay? So how much carbon dioxide is in our blood, or hydrogen's in our blood, or oxygen, or nitrogen, okay? So you have chemoreceptors that will tell, all right, how much of a specific type of chemical or how little of a specific type of chemical is present in a fluid, okay? We're going to talk about today when we olfaction, okay, smell of food, okay? Chemoreceptors are involved in our sense of smell and taste, right? The next is thermoreceptors. I have a feeling I don't need to explain that to you folks. Thermo, temperature, okay? Obviously, we have tons of thermoreceptors in our skin, but also we have some in the hypothalamus because our hypothalamus regulates our body temperature, right? Which is always fascinating to me because the hypothalamus is what spikes a fever for you. It hooks you up. And part of that, all right, it's not always the case, but part of that is if you turn up your internal temperature by a couple of degrees, certain pathogens can't handle it. Certain bacteria don't like it. It becomes a hostile environment, and you can prevent them from reproducing. And in some cases, you can kill them off. And it's just as little as turning up your body temperature. It's cool. I took a micro class once. Did you guys ever take micro? Do you have to take micro for anything? No? Okay. All right. It's a tough class, but I don't eat the same after taking the microbiology class. And I overcook all my meat. All right. The next type of receptors, photoreceptors. All right. This is in the eye. And you definitely need to know this. We won't talk about it today. We'll talk about it in a later class. All right. Photoreceptors are going to detect changes in how bright or how low lit something is. So light intensity, the color, and movement. Okay? These will be found in the retina, which is the neural layer, the inner layer of your eye. All right, 2 more. All right, mechanoreceptors. Okay? These guys, all right, proprioceptors kind of fall in with mechanoreceptors, right? But these receptors are going to be found, all right, within cell membranes. And so when the cell membrane becomes distorted, for example, if you touch something, the cell membrane becomes distorted, and these receptors pick up on that, all right? So they're going to include touch, pressure, vibration, and then, hey, there's our stretch receptors, right? The muscle spindle. So baroreceptors detect stretch. Proprioceptors, we know what they detect. Tactile is touch, okay? And then we'll get into the specialized receptors in the inner ear. Those are pretty cool, the hair cells and whatnot. Those are all mechanoreceptors. And then finally, last but not least, nociception. All right, the nociceptors, pain, okay? And there's 2 types, somatic and visceral. All right, visceral is easy. That's going to be found within the internal organs, and they monitor if any of those organs are damaged. All right, somatic, all right, this is what we're going to see on the surface like our skin, all right, also found in skeletal muscles. So they're going to detect damage if it's chemical, heat, or mechanical. Okay? Nociceptors are quite important. All right. So receptors on the tongue are classified into what three receptor criteria? All right. First is the tongue is part of the special senses, taste. Okay, and taste receptors fall under the special senses because they're involved with the head. Okay, they're exterior receptors because when you're tasting something from outside your body is being brought in, all right, into your mouth. Snakes do it all the time. When they stick their tongue out in the air, all right, they're able to sense the external environment. So they're using their tongue. They have receptors on their tongue that can help them understand the exterior environment. And then chemoreceptor, all right? And we'll talk about that later on, but remember, the chemoreceptors have to do with chemicals that are dissolved in a fluid, your saliva, okay? All right, I'm going to kind of run through the tactile receptors here. I don't want to say quickly, all right, but because this stuff usually I lose students, they get a little bit bored here, all right. But tactile receptors, these are the touch receptors, all right, and they're going to be found, all right, in your skin predominantly, and then the mucous membranes, okay. And there's two types: encapsulated and unencapsulated. So we're going to start off with the unencapsulated tactile receptors, and basically what that is that the touch receptors, right, have dendritic nerve endings here, okay, that come off. You know the dendrite there, okay? But they don't have a protective cover. So when we get to the capsulated ones, those will have a protective cover, but these don't. All right, the first type are free nerve endings, okay? These are commonly seen, all right, for pain and temperature, all right? That's where you'll see them the most. And then the most basic type, all right, of tactile receptor, all right? So you'll find most of these in the skin, also within our mucous membranes though, right? And they can be either phasic, all right, or tonic, right? So they can either be slow to adapt or quick to adapt, okay? If they're phasic, adapt rapidly, right? Which is good because then they're not going to decrease the sensitivity, all right? That's your pain receptors, all right? Tonic, they'll adapt slower, all right? So they'll decrease their sensitivity, all right, much more quickly, okay? Hair root plexuses, again, these are going to be found, all right, down in the dermis there, okay? We talked about these before. And so when you're outside and the wind blows across your arm, okay, and you can feel that, all right, on your skin, but also when your hair is moving, all right, it's moving the hair follicle, all right, and these hair root plexuses, all right, are attached to the hair follicle. So you can feel that it stimulates those, okay? So that they detect that hair displacement, that movement there. Someone asked me once, when you're getting hair plucked, do those sense? I said, I have no idea. I don't know. I couldn't tell you. All right. And then finally, we've got our unencapsulated tactile discs. All right, we saw these before when we were labeling in the skin. The tactile cells, remember the Merkel cells here? All right, these were those guys that sat there in the stratum basale, that basal layer there. All right, and so they're the epidermis, that's the outer layer of your skin. Okay, because remember, your epidermis is made up of epithelial tissue. Okay, you don't have any sensory receptors in there. All right, you have this specialized cell here. All right, this nerve cell here. All right, and so they respond to light touch. Okay, it is the closest to the surface. All right, let's jump into the unencapsulated. Well, here, I skipped over the picture. If you want a quick picture here to see, all right, what these guys look like. Okay, here's the free nerve ending. Majority of these are going to be like pain receptors. Okay, temperature receptors. All right, here's your root hair plexus. See how it wraps around the follicle here? So if the hair moves at all, it stimulates this. And then we saw this back in chapter 6. There's that tactile cell here. All right, and that tactile cell, when pressure gets applied to it through light touch, it deforms the plasma membrane here, which stimulates the tactile disc here, which will then send that sensory information to the brain. All right. The other type of receptors for touch are the encapsulated ones. All right. And by definition, the encapsulated ones, all right, will have a protective covering. All right. Connective tissue or glial cells. All right. In the periphery, it's going to be neurolemmacytes. What's another name for a neurolemmacyte? Schwann cell. Remember that? Schwann cell. All right. So the endings for these neurons will be wrapped in either connective tissue, okay, or glial cells. All right. So the first type, end bulbs or Krauss bulbs, all right, these guys again will be located in the deeper layers of the skin, also in the mucous membrane. So they're going to be in the deeper layers. They're going to be associated with pressure and low frequency vibration. The lamellated or Pacinian corpuscles, all right, we saw those when we were labeling the skin. That's the one that looks like an onion. Remember, it had that little stalk there, and then it had all these layers. Well, for a moment there, I was really hopeful this was going to turn into a good drawing. All right, but it's like an onion, okay? So it has these layers to it, okay? And we saw that. That was located deep in the dermis, all right, also in the hypodermis, all right, which is even deeper. Okay, so how I remember what this monitors, because it's deep into the dermis, it's deep pressure. Okay, and you'll see it inside, and some of these will be inside the organ walls here. So coarse touch, unrefined touch, all right, and then high frequency vibration. Okay, those are the encapsulated ones. All right, 2 more to go. And then I promise you we'll get to some cooler stuff. All right, you got the bulbous or Ruffini corpuscles. Okay, again, these will be in the deep dermis and in the subcutaneous or hypodermis. So again, this will be deep pressure. And then finally, our Meissner corpuscles or tactile corpuscles. All right, we saw this in the dermal papillae. Remember those little structures that help to give us our fingerprints? Okay, the epidermal ridges and whatnot. Well, the dermal papillae, okay, will have these guys in there, and this helps with discriminative light touch. Okay, so when you have your eyes closed, and I ask you, all right, open up your hand, and I put a distinguishable object in there like a key, all right? If you can feel that, you're like, oh, all right. Or if I put in a lucky rabbit's foot or something, you can say, oh, that's soft and furry. All right, that's discriminative light touch. So a quick picture. All right, there's the end bulb there. All right, then you got your lamellated or Pacinian corpuscle, all right, your bulbous corpuscle, all right, and then finally our tactile corpuscle, our Meissner's there, and that little dermal papilla here. All right, now that wasn't too bad. I remember teaching that to a class once, and people were just glazing over. All right, referred pain. Who doesn't like referred pain? Well, I don't if I'm having a heart attack, but referred, or if I have appendicitis. But now we're going to learn about what the heck causes referred pain. How come, you know, when I'm having a heart attack, that my left arm is having pain in it, in my left chest, all right? Why is that happening? Well, let me tell you why, okay? So what happens is that your body has a hard time localizing, all right, where all the sensory input is coming from, okay? Because a lot of time, visceral, your organs, all right, and the sensory neurons that are affiliated with certain areas of your skin and certain muscles, they all share the same pathway up to the brain. Okay, it's like a whole bunch of people, all right, riding the same elevator to the top of the building. All right, if all of us were to get on there, we're all different people. All right, we might smell different. All right, might talk different languages. All right, but we're all going to the same place. All right. So that's what's going on here with referred pain, okay? So some of our somatic and visceral sensory nerve endings are all going to travel in the same ascending track. Remember, an ascending track is a sensory track, okay? And it's going to travel in the spinal cord up to the brain, okay? But the problem is, all right, the cerebral cortex cannot figure out, all right, where this information came from. Does not know. Okay, so this picture down here, is showing us that, right? So you can see here's this area of skin surrounding the umbilicus. It's called the periumbilican region there, okay? And you've got a bad case of appendicitis, okay? So that region of skin and the appendix both take the same elevator, the same pathway up to the spinal cord. So you can see you've got this sensory information from the somatic sensory neurons traveling along here into the spinal nerve, and then you've got the visceral, all right, information, sensory information from the appendix. It's going to ride along the same spinal nerve, right, into the spinal cord and go and ascend up to the brain. So it's getting information from both places, but it has no idea where the true source is coming from. That's why when the appendix is inflamed, you can initially get periumbilical pain, all right, around your belly button, but then it migrates over to the right side of your body there. Same thing, all right, with a heart attack, okay? So the heart, all right, gets sympathetic innervation from T1 through T5, all right? But guess what? The skin segments, all right, to those regions include your chest and your left arm, okay? So that's why when you get a heart attack, all right, you have pain in both your left arm and your chest, obviously over the heart too, all right, because they're taking the same pathway up to the brain. All right, kidney and ureter pain, all right, share T11 or T10 through L2. That's going to be more in the low back. Has anyone here ever known anybody to have kidney stones? All right, very painful, very, very painful. If you're a man, it's the closest thing. They've said this, that the closest thing a man will ever know to giving birth. I don't know, I couldn't tell you. But anyways, all right, so we'll see, all right, the whole point of referred pain is, all right, you will have specific areas of your skin, okay, which are going to share the same pathways, all right, those same sensory pathways as visceral organs, okay? So this little map here shows you, all right, we're all familiar with the heart one, okay? Left pectoral region, all right, left arm, okay? Liver and gallbladder, right shoulder, right, most commonly. And if anybody has ever had gallbladder pain, it's a little bit more common nowadays. Usually they'll complain of right shoulder pain. It's almost guaranteed, all right? Now, it's interesting because when I was in school and we were learning common sites for referred pain, the pancreas was usually right in the middle. This book, this publication, has moved it off to the left a little bit. It varies, all right? Point being is, all right, you will see that you'll have different regions here, all right? Kidney, low back region here, all right? Ovaries, pretty general, right where the ovaries should be, okay? Appendix, periumbilical. How about phantom pain? Folks heard of that? Someone missing a limb, okay? But yet they'll still feel pain in that limb. Well, the wiring is still there. Even though you lost your arm, let's say, or your leg, okay? The wiring in your brain is still there, okay? So every once in a while, all right, those nerves will get stimulated. Those sensory neurons get stimulated because that hard wiring is still there in your brain, and you'll feel discomfort. And at times it can be excruciating pain. It can be really, really rough, all right? But even though we have actually taken that limb off, right, the cell body is still in existence in your central nervous system. And every once in a while, it will stimulate, all right, the cerebral cortex and give that person the sensation that the limb is still there, all right? And in some cases, it could be quite excruciating. All right, let me jump into smell. All right, now we're going to get into, folks, this is it. We're going to just move through our special senses here on out, okay? So we're going to start off with smell, then we'll get into taste. And I'm trying to think. I think it's vision, then hearing, then balance, okay? Equilibrium, okay? So I talked briefly last class about smell. We're going to get into a little bit more detail. So first things first, all right? When we talk about olfaction, okay, we're going to be dealing with chemoreceptors, all right? And keep in mind, all right, these chemoreceptors are going to detect chemicals, all right, dissolved chemicals in a fluid. But these chemicals, all right, are derived from molecules from the external environment, the air around us, okay? So these molecules, we give a name. We call them odorants, all right? Now, the book describes them as volatile molecules. And usually when I've come into contact with the term volatile, I think of things that explode. They don't, all right, in this case. Volatile molecules are just reactive molecules, all right? So we call those odorants, all right? And these odorants come from the external environment, all right? And they make their way into your nasal cavity. And then they actually embed themselves into the mucous membrane of your nasal cavity there, okay? And in that mucus, all right, we have our chemoreceptors. And so they're going to give us information normally about food for the most part, but we can also tell if people smell bad. Horses, I don't think all horses smell bad, but you know what I'm talking about, all right? And then also in some cases, they say you can smell danger. I don't know. Maybe I'm just not in tune with that. But in some cases, you ever seen those movies when like the farm animals start freaking out when they know something's up, you know? Dogs can smell danger or whatever because they can smell predators. I don't know. I've never been able to smell danger. But if you watch Jerry Maguire, the little kid says that dogs and bees can smell fear. So I guess dogs can smell danger. Okay, so we are able to actually smell many, many different things, okay? So not as much as certain other species, but we can, all right? And in some cases, we probably wish that we couldn't, you know, if it's a bad cook or whatever. But inside of our nasal cavity, all right, the lining of the nasal cavity is lined with epithelium. We refer to that epithelium as the olfactory epithelium, okay? And if we go back to this picture here, which I showed you before, all right, our sensory receptors are going to be in the superior region of the nasal cavity, all right, up there. That's why it's always encouraged that folks, if you really want to be able to smell something, you need to take a deep breath to get those odorants up there, up towards that mucous membrane, up towards the superior portion there, okay? So the three types of cells that we're going to discuss in that olfactory epithelium are, number one, are olfactory receptor cells, okay? They have one job, to smell everything, okay? The other type, or the second type, is the supporting cells, and their job is, they're like the glial cells for the neuron, okay? Their job is to sustain, maintain, support, protect the olfactory receptor cells. And then the basal cells, I always think of them as baby cells. All right, they're going to take over. Okay, so our olfactory receptor cells don't have a very long life. All right, about 40 to 60 days. Then they turn over, which is good. Okay, because old cells, as you know, throughout our many chapters here that we talked about, as we age, things don't work as well. All right, but our olfactory cells don't have a very long life, so not a problem, because when they, you know, take off and die, we've got these basal cells to move in and take over for them, okay? Okay, so one of the things that you need to understand, that our olfactory epithelium sits on this tissue layer called the lamina propria here. And it's a loose connective tissue, all right, which is loaded with blood vessels, nerves, and most importantly, our olfactory glands. And it's those olfactory glands, okay, which are going to support that mucus layer. So those odorants can then come in, embed into the mucus layer, dissolve into the mucus layer, and in the mucus layer is where we're going to have our cilia, all right, which are going to be those receptors at the end of the olfactory neuron there, right? Let me jump in and show you the picture here. You might understand it a little bit more, okay? So here we got our friend here smelling the coffee, hot cocoa, all right? The odorants are now traveling into the nasal canal, nasal cavity, swirling around, and then they're going to embed themselves up here, all right, into that mucus layer, which is right here. Okay, so our odorants then embed themselves in the mucus layer, which is made, all right, by the olfactory gland or the Bowman's gland here in the lamina propria. Okay, and also in that mucus layer are going to be the cilia or the olfactory hairs, which have the receptors there. Okay, so if this neuron has receptors that can detect the odorant, then you'll get a nerve signal. You'll get a nerve signal that will be transmitted up to, all right, the olfactory bulb here. Okay, so here's our olfactory epithelium. You can see the three types of cells. Olfactory neuron, all right, you've got your supporting cell here, these long cylindrical cells here, and then you've got your baby cells, the basal cells. They're just waiting, which sits on this layer, the lamina propria. That's that loose connective tissue loaded with blood vessels, all right, the glands here, and then nerves. And then that all sits on the cribriform plate of the ethmoid bone. On the other side of it is the olfactory bulb. And basically, the olfactory bulb is just where a bunch of synapses are occurring between the olfactory cells here and these mitral and tufted cells. Okay? And then from the olfactory bulb, all right, we will get the olfactory tract. I'll talk about that in 1 second here. Let me just pull this up real quick. Okay? So a couple things about the olfactory receptor cells. One, it's a bipolar structure. Okay, so we have our cell body at one end. At the other end, we have the dendrite with the cilia. Okay, and then the other end, we've got the axon that's moving up towards the olfactory bulb here. Okay, so it's that cilia that has the receptors, all right, for our olfactory receptor cells, and that's where those chemoreceptors are going to hang out. All right, and if that odorant is specific for that chemoreceptor, it will activate the chemoreceptor. If it doesn't, you won't smell anything. All right, that's why dogs can smell better than us, because they have many more chemoreceptors, all right, for many more types of odors. All right, and there's certain things that we just can't smell. Okay, we can smell a lot, but there's still some things that we can't smell. All right, please don't forget that your olfactory nerve is cranial nerve number 1. All right, you only have one nose, okay? All right, so from there, all right, we are going to, well, I already talked about that. Our olfactory bulbs, they're a pair, you have a left and a right one, all right? From the olfactory bulbs, we then have our olfactory tracts, and they run on the underside of your brain on the frontal lobes there. And so they'll make their way all the way back, okay, to the temporal lobe, where you have your primary olfactory cortex. But they'll also have connections with the hypothalamus, the amygdala, all right? Hypothalamus and amygdala, that has to do with the limbic system, okay? But also the hypothalamus has to do with visceral responses. But a very important point on the slide is this guy right here. And I said this in the lab last week. Okay, the olfactory sensation does not go to the thalamus. Okay, it is the only one that does not go to the thalamus. That's a great true-false question. I hope none of you missed that. I know none of you will. Yes, ma'am. Yeah, exactly. The habenular nuclei. Yes, the habenular nuclei is in the epithalamus. Remember. Well, it just doesn't, it's interesting you say that. Another student that sat in that exact same seat last night asked me the same question. And it's not because of that. It's one of those sensations that doesn't have to ascend up to the hypothalamus because of its location. It just bypasses that. So basically, how do you smell something? This is the last slide, and then we'll take a break, okay? Take a deep breath, sniff something, whatever. Circulating those odorant molecules, all right, throughout the nasal cavity, they'll come in contact with what we call odorant binding proteins, okay? And it's these proteins that are located in the mucus here, all right, they'll bind on. Now, this is important because you'll talk more about this in detail in 211 because you'll talk about secondary messengers, but that's what this basically involves, all right? These odorant binding molecules here, all right, are going to stimulate, all right, those chemoreceptors, all right, to initiate a G protein, okay? It's a secondary messenger. Well, basically what that is a G protein lives on the inside of the cell membrane, okay? And that G protein, all right, is going to activate this enzyme, adenylate cyclase. Remember, AS, that's an enzyme, all right? And basically what that does is it converts ATP, okay, into what we call cyclic or C-AMP, cyclic AMP. And then what that cyclic AMP does is it's going to cause the opening of sodium and calcium channels. It's going to cause depolarization. Because remember, we need to create a nerve signal. We need to depolarize that cell. Once that occurs, we've triggered an action potential because we reach our threshold value, and then our nerve signal is going to be conducted down the sensory neuron pathway. Okay? So some of these other structures that are going to be involved is obviously the cerebral cortex, because this is olfaction, so that's going to be in the temporal lobe. So you can actually identify what you're smelling, and you can actually have that sensation of smell, right? But also, you ever notice when you smell something, right, that if it's something that you really like and you're hungry, okay, it can cause you to salivate? Well, that's because you're stimulating the hypothalamus. All right, and you're going to start to get the visceral response. You might even get some gurgling in your stomach because your hypothalamus is stimulating secretion of some of the digestive chemicals in your stomach to begin, all right, digestion. Even though you might not eat, but you still get that response going on. All right, so that's what the hypothalamus can do. Now, if you ever smell something, you're like, oh, I don't like the smell of that. That's an emotion, right? So we are triggering. I can't talk today. We are triggering the amygdala. Okay, but if it's something that you like, you're like, oh yeah, you know, this is good. I'm really happy about that. Same thing. You're triggering their amygdala, and you're going to get that emotional response through the limbic system. Okay, so that's why, you know, the olfaction doesn't go just directly to the temporal lobe and just quit. All right, that pathway is going to actually hit a couple of other areas, especially because if we're going to start to eat, all right, digestion, we got to get everything ready. We got to get your mouth ready. We got to start to salivate. We got to start to get that process ready as you start to chew on things and swallow food. And we got to get your stomach up and running because we got to start digesting some stuff. So we have all these connections there. All right, we didn't make it as far as I wanted to. Actually, we did, right? But I kind of wanted to do taste too. But this is good. This is really good. Let's take a break. I know I was talking.